Hey, AP Bio folks. Hope you guys are all staying healthy and happy. Excuse me. Um, you'll notice a different background for the rest of my videos for the next few weeks because I have COVID. So now I am in quarantine in my bedroom, away from my family, and I'll be coughing a lot. So I apologize for that. But sorry. Um, anyway, I wanted to make a video to go through the notes on oxidative phosphorylation. So if you guys would make sure you download page 37, the oxidative phosphorylation skeletal notes will probably take about 20 minutes to run through them. And then that's sort of our major topic for the week. And then I have a bunch of other activities and things to, excuse me, to go through and make sense of it all with you guys. So make sure you have that downloaded and we will take a look, all right? I'm going to take myself out of the video here because you don't need to stare at my headboard on my bed. So here we go. If you continue from what we said on the last video set, the excuse me, the idea here is that we can understand this idea of oxidative phosphorylation, which is basically what happens during the electron transport chain and how it leads to ATP synthesis. So we've got to look at just a handful of things about it, and a lot of it right here is going to be dealing with math and how we know where the different bits come from, how much ATP is produced where. Excuse me. And then after we've got that all figured out, we're going to take one last look at fermentation and anaerobic respiration. So we'll do a quick little review of that. And then lastly, we'll look at some other metabolic pathways that could happen, which is the real last part of the chapter. So here we go. We'll take a look at it. So far, what we've looked at is these ideas of what are the inputs and outputs that go into and out of the initial parts of the process, right? So far we know that we always need to start with glucose. We break it down via this act of glycolysis to create two pyruvates. It also creates, during that process, two ATPs that we have available, and then we make two NADHs from that breakdown. Now, the glucose directly breaks down into two pyruvates. So those two pyruvates go through this right here would be what I was calling a link reaction. And then from that link reaction, you get two acetyl-CoAs. Those enter the citric acid cycle, excuse me. And as you go through that cycle, it happens twice because there's two pyruvates. So you go through it twice, and then from that circular motion, you get one ATP each time, so a total of two ATP. You get 8 NADH, you get 4 the first time and 4 the second time. You get a total of 3 CO2s the first time, 3 CO2s the second time, so you get all 6 of them there. And then you get 1 FADH2 the first time and a second the second time. So if we're looking at all of our math so far, what we've produced up until where we stopped was 4 ATPs, 10 NADHs, all 6 waste product CO2s, and then 2 FADHs. So now what we're going to focus on is we've already used that, and that's already gone. So we're going to look at what happens with all of those and where do they go. Excuse me. So that is our goal. So we're going to have to do a little bit of energy accounting on this one. So when you look at it, by the end of glycolysis, Link's and Krebs cycle, how many of the original 6 carbons have been converted to 6 CO2? Well, you guys just said it, but all of them have, right? Which would be 6 CO2s. How many ATPs have been produced at this point? Right, we've only made 4 so far, and they are all through substrate level phosphorylation. We haven't used oxidative phosphorylation yet at all. And then the other question, so where is the rest of the energy that was originally in the glucose? It is in that NADH right? We have 10 of those, and it's in those two FADH pluses, or FADH2s. I always think of those things right here as these, as big old piggy banks, right? So they are sort of stored energy that we still can break into. They are, remember, they're electron carriers. So the electrons that are found in here and the electrons that are found in here, we're going to use those to make our way to make more energy. Okay, so that is what we want to look at. So if we look at this thing, so far the ATPs we have been generated via, and I just said on the last slide, but let's clarify again, that substrate level phosphorylation. Right, remember that's the where basically a substrate comes in, it has a phosphate removed from it, it's added to ADP to make ATP. That's what we've done there. So now it's time for what we call chemiosmosis. And that is what happens in the electron transport chain, right? So, excuse me, guys, I'm sorry. Where is the energy coming from for the active transport? So where is that energy coming from? Right? Well, it's got to come from those things that we produced in glycolysis and that link reaction and the citric acid cycle. So it's coming from the electrons that are stored in FADH2 or NADH. So that's where that energy is coming from. All right, so what we're going to do is send the electrons that are stored in those into the electron transport chain, and then through that process, as that electron moves down, 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 it's going to move all these hydrogens through, create a massive gradient up here, right? That's that inner membrane space in the mitochondrial membrane. Then all of that gradient is going to go back through ATP synthase and produce ATP. And that's, I mean, realistically, that's all it is. So it sounds a lot more complicated. It looks a lot more complicated, but in reality, it's actually relatively simple. I'm gonna put a link. I love this video. I think once you learn the process, you can go back in and click on this link. This is a little rap video produced by, I believe it's Stanford. I think it's Stanford, but a college. They make a little video about the process of a little rap video of oxidative phosphorylation, which is a pretty good one. So I'll put a link to it. That's what this slide was about. So I'll have a link there listed for you. You can go back and check it out. But, excuse me, what pulls the electrons out of the chain? That's the question here. So basically, if you look on here, I don't know if you guys ever saw the Sunday morning cartoon Mighty Mouse, but Mighty Mouse here is the electron that's going to get pushed all the way through until he has that look on his face, which means I've done all the work I can do. I'm now stuck. Okay, this is the electron transport chain. So there's a series of enzymes here. They're numbered 1 through 5, or 1 through 4. And then there's a fifth one, which is the ATP synthase. We're not showing you that one on this image. But basically, that electron, as it gets there, so for every NADH, 2 electrons are going to get pulled out of it at this first protein, and they're going to make their way through. And as they do, they're going to move hydrogens through. Right? That's the whole point. FADH2 doesn't carry quite as much energy as NADH, so they're going to actually join at the second protein. And they're going to drop off their electrons, and they're going to go through. Now, as they go down this conveyor belt, they get to the back here, and then this one gets to the back here, and then another one would come in, and another one, and they basically would back this whole thing up. Much like I've given the example before, if you're at Cub Foods, bagging your groceries, and you can't keep up, the conveyor belt stops because it's too full. Well, that's what happens in the electron transport chain. So something right here has to clear those electrons out so that this thing can continue. So we're asking is what pulls electrons out of the chain? And the answer is oxygen. Oxygen is the final electron acceptor. So it pulls those electrons out to make water. Remember, water is that other waste product. So oxygen bonds with some of the hydrogens that are in there, excuse me, and those electrons, and it makes water. And then that clears the system right here so that the next round can happen, and it keeps it going. What happens to the energy that is lost from the electrons? Well, as that electron starts out big and powerful and it works its way down till it's little and it's lost all that energy is moving electrons against the concentration gradient. Right? Hence, it's active transport. So it's trying to build a gradient, and you'll see why here in just a second, but that gradient is ultra important. So if I said to you guys, let's draw it, I'm not going to go crazy here, but basically, if you look at the mitochondria, right, it's got this folded inner membrane. Okay, so where we're looking, excuse me, where we're looking on here would be on this portion of the membrane. Right? So if I was to draw a membrane, I'm drawing just this inner membrane. So there would be, right, here's the membrane. This would be the inside here, would be down here. This would be the inner membrane space. And then there would be an outer membrane out here, right? But in here, what we would have is we'd have a series of these proteins in the electron transport chain. We would also have, and they have names, but for now we'll just say this one's 1, this one's 2, this one's 3, this one's 4, and then there's a 5th one. It looks like a big kind of cannon, almost like a skull if you put some eyes on it, right? But that one looks like, this one is ATP synthase. The rest of them, excuse me guys, the rest of them are here to work and move hydrogens across. And then there would actually be, I'll put it in blue here, there would be 2 small ones, and their job is to actually go back and forth and move that electron down the pathway. So basically, if I use blue, so if NADH comes in, it comes in right here. It turns back into NAD+. It releases that hydrogen and that electron. That electron is now powerful. So as that electron gets moved all the way through here, it's going to take a bunch of these hydrogen ions and it's going to push them up here. And then FADH comes in, right? Turns into FAD, has some H's. Those get pushed through here, So we create a huge gradient up here of H's. And they are all moved up there by the electrons from NADH and FADH2. Now, once we've moved that up there, we create this high concentration up there. Now they go through osmosis, basically, diffusion. This is the whole idea of chemiosmosis. These are going to come flying back through here. And as they do, ADP is going to get changed into ATP by taking a phosphate and slamming it onto it. Basically, this thing rotates almost like a barrel of a old 6-shooter kind of pistol from the old Western movies. Kind of spins like that, and it puts the phosphate on there to make ATP. And the more of it that cranks through there, it makes more and more ATP. All right, I know that's a pretty weak drawing, but I have a better version that I'm going to ask you guys to kind of review with here as an assignment this week. So it'll get you a much better drawing. Okay. And again, if you have questions, obviously this week, this time that we have in our meetings this week, our Zoom meetings, I assume we're going to have a lot of questions to go over with this. So jot those down and come to Zoom meetings with those ready to go. Excuse me. But if you look at the efficiency of ATP production here, basically the book gives you a couple numbers here so that I'm going to ask you to kind of know. they're saying for every NADH, it makes about 2.5 ATPs. For every FADH, only 1.5 ATPs are made. And that's due to where they actually come in to the electron transport chain. Remember, NADH drops off at that first protein. FADH2 drops off at that second protein. So it has a little less, can move a little less hydrogens. Now, if we know that we have 10 NADHs, right? If we have 2.5 per, that makes us have 25 ATP produced. If we have 2 FADH2s, which we do, and we know those make about 1.5 each, that gives us about 3 ATP. So we know that if you add those up, we get 28 ATPs are going to be from oxidative phosphorylation. Right? We made 4 from substrate level phosphorylation. So 28 plus 4, so there we have our 32. Okay? If we look at the numbers here, and again, each book you're going to see different numbers. We said you might have, some books will say 36 or 38. We're going to kind of look at these numbers here using these ones. We'll say, what is the efficiency if glucose is free energy, right? This is that whole free energy idea. If glucose is negative 696 kilocalories per mole, and the ATP is the free energy of ATP itself is negative 7.3 kilocalories per mole, right? So basically, how efficient is your body at producing ATP? Well, if you think about it, if I say negative 7.3 times the 32 that I have, right, that equals, what is that, negative 233.6, I believe, and I divide that by, right, what it is for the glucose itself. I take that times 100. It gives me about 34% efficiency. So that is as efficient as your body is. 34%. That's not great, right? But it does give you all the energy we need from food to do our business, right? So you think of it as not very efficient, but in reality, that's a pretty good number for us. It's a pretty good evolutionary adaptation that we all have to be able to do this. Excuse me. So, excuse me, yikes. When you look at this we already saw this one before, but if you go through this and you look at what we have sort of talked through, all right, you can fill this in, but these are your glycolysis is here. Oops. This is your glycolysis. Right, this is that pyruvate oxidation or that link reaction. Right, your citric acid cycle. This is your oxidative phosphorylation. It's your ETC plus your chemiosmosis. Right, so if we know the general things here, these boxes then we get, these have to be the different NADHs, right? These have to be the NADHs. These have to be the NADHs and the FADH2s, right? These upper ones here, as they get shuttled in, it's just showing you that as they come in, it's still these NADHs, or there's a potential having FADH2s. And then we know we've got our 2 ATPs, and we've got 26 to 28 ATPs. So the number here will be somewhere in that realm. All right, that's kind of what our book goes with. So I did the math out at 32 when I did it for us. So when you think about or look about some of the ideas here, one of the things the chapter talks about is what these oxidizing agents. When you look at it, the big one here is right here, the oxidizing agent for glycolysis. In other words, what basically drives this reaction, right? Oxidation is a loss of electrons, right? It oxidizes it. Reduction is a gain of electrons. It's a little weird. You're reducing something when you gain electrons because you're reducing the amount of positive, all right? So in this case, what we're looking at for oxidation during glycolysis or the Linker reaction or the Krebs cycle is NAD+, right? So the answer for this one is NAD+. The answer for this one is NAD+. The answer for this one is NAD+. Why? Because NAD+ is the thing that is accepting those electrons. So it's the oxidizing agent. It's accepting those electrons to make it NADH. How is the NADH obtained to run all of these reactions? Well, this NAD plus is coming back after it's been emptied during the electron transport chain. It brings the NADH, it takes the H's out and sends it back as NAD+, so you can reuse that again. So it's actually being returned from the electron transport chain. So you can see sort of the cyclical nature of this thing. Right, oxidation of NADH and FADH2 via electron transport chain. That's the answer for this one. Should be bullet pointed in. Without oxygen, what happens to the regeneration of these molecules? Well, remember, oxygen is the final electron acceptor, so it pulls out those and allows this process to continue. So without oxygen, we would be able to do this for just a little bit, and then the electron transport chain would come screeching to a halt because oxygen couldn't remove those electrons, and the process would stop, and we'd have no more, we wouldn't have any more of this being converted back into this, which means we couldn't drive this process. So basically, everything would get stuck in this, and we'd come to a halt, right? So we need a way to have oxygen to pull it out. Well, the question then can be, well, what if we don't have enough oxygen, right? How can we continue this process? Because we need to. We can't just let it come to a stop and not produce anything. So right here, if we don't have oxygen clearing this out, right, to make that water, how can we continue if that oxygen isn't present? How can we keep glycolysis going? And the answer is fermentation, right? And that's the key. So when you look at this, what I'm showing you in these two pictures here is two different types of fermentation. So both of these are versions of fermentation, which we know is anaerobic. It doesn't require any oxygen. What you'll notice is this first bit right here and right here, they're the same, right? We go from glucose, we do glycolysis, we create these two pyruvates, right? We've created a little bit of ATP, but if there's no oxygen, these pyruvates aren't going to go continue to the Krebs cycle. So what they have to do is they have to have an alternative pathway. And what they're going to do, because we've already filled up these NADHs, so basically we have to, because we don't have the electron transport chain to empty these, we have to use a different process. So instead of taking that pyruvate and kicking it into the Krebs cycle and continuing to make lots of ATP, we're stuck. So what we do is we take that NADH and we empty it out to make NAD plus again, which can then at least go back and make those two ATP. So at least we're making something. Both of these are going to make just that little bit continuously, right? But what we've done in the process, when we've emptied that NADH, we've made a couple other things. So in this case, we make ethanol. And so this would be alcoholic fermentation. So we do not do this in our bodies. This would be something like yeast will do this one. We do what's on this other side, right? So what we're making here is lactic acid, right? So this one is lactic acid fermentation. And that's what humans do when we have lack of oxygen to continue the process. What has more potential chemical energy, pyruvate or lactate? Well, they're actually kind of the same, but we have to, well, pyruvate has more potential because we have that to go into the Krebs cycle. So at some point, once oxygen becomes available again, we have to be able to convert this back into pyruvate so then it can go on to the Krebs cycle. So fermentation sort of has this alternative pathway to keep us working just a little bit at least, all right? So, excuse me. So what I'm showing you on this one, I pulled this out of your text as well, but right here, this would be our pyruvates that we create, right? We create 2 of them via glycolysis. If no oxygen is present, then we do, depending on, so we're talking humans here, we would do we would create lactic acid. If we were talking about some bacteria, you could be creating ethanol. If oxygen is present, then we make acetyl-CoA, which goes into the citric acid cycle and continues. So this would be our sort of delineation of oxygen or not. Notice right here, then we just kick this back this way. Here, we have to finish the whole electron transport chain, so we're making a lot more goods to go along with it. Now, excuse me. In your textbook here, this image is from, it shows you some alternative pathways which we don't study quite as much, but we always think about this center one right here. Okay, we have carbs, that's our sugar that we're going to break down, it goes through glycolysis, glucose turns into pyruvate, goes through acetyl acid CoA to the citric acid cycle to oxidative phosphorylation. So that's the one that we want to typically use. But you can also see that if you have proteins that you need to break down, they actually will skip and join farther down into the process. So they're not as efficient at making ATP for us than other things. So proteins and fats both can, right? They can jump in and produce energy as well, but they sort of have a slightly alternative pathway, which we don't have to get into too crazily. All right, last couple slides here. In this case, I'm asking what would be a good allosteric or competitive inhibitor. Remember, in biochemistry, an allosteric or competitive inhibitor, it's the regulation of an enzyme by binding an effector molecule at a site other than the enzyme's active site to kind of shut things down, basically. So in this case, right, what is going to be the inhibitor to this? It's going to be ATP. If we have massive amounts of ATP, it allows us to sort of stop the process because we don't want to create bundles and bundles of unusable ATP because we don't have anywhere to use it. So if we have too much of it or we make a lot of it, it'll inhibit the process from happening as quick, right? So it would slow it down. So I'm trying to show you guys some of the biological controls for this. So some other options to look at. So basically, this is a sort of a FRQ type idea. I already drew the line in here for you, but it says this graph shows the pH difference across the inner mitochondrial membrane over time in an actively respirating cell. At the end of the time indicated by the vertical arrow, which is right here, the metabolic poison is added that specifically and completely inhibits all function of the mitochondrial ATP synthase. Draw what you would expect to see over time. So this, if right here you don't have any more ATP synthase using, it's not going to clear those hydrogen ions that have gathered in that intermembranous space, and the pH is going to change drastically because those H's are going to be stuck. And remember, that's creating a very acidic environment in that inner membrane space. So it kind of just a heady question, sort of, to think about. But we can talk about this in class in our Zoom meetings. That is the last slide I've got for you. I apologize for for coughing, but getting COVID is not fun. It's like a nasty flu that just does not go away. So hopefully you guys are staying happy and healthy. And if you have questions, come to class, come to Zoom, ready to chat and ready to draw. So we'll do some drawing and sketching and try to make sure everyone's comfortable. This one, I'm not going to use breakout rooms. We're going to stay together and we'll do a kind of a question answer kind of thing. So have a good one, guys, and we will see you in the Zoom meetings.